Good afternoon and welcome to the North American Vascular Biology Organization's 7th webinar. I'm Linda Shapiro from the University of Connecticut and a member of the NAVBO Education Committee. I will be monitoring today's session. We're pleased to welcome our speaker, Ann Eichmann of Yale University, who will present her work entitled Lacteal Junction Zippering Protects Against Diet-Induced Obesity. Much of this presentation will refer to her article of the same name in Science. A link to the abstract of the article was included in the program materials. If you have a subscription to Science, you'll have access to the full text. Before we get started, I wanted to go over some logistical aspects. Throughout this webinar, you're able to switch between the phone audio and computer audio in case you're having a problem. You can see this information on the audio section of the GoToWebinar control panel. If you experience technical problems, please click on the Help tab at the top of the control panel. Scroll to the bottom of the help screen for the technical support phone number. At this time, I'd also like to welcome Dr. Gael Genet, also from Yale University and a co-author on the paper. He will monitor today's questions. Questions will be handled in two ways. Throughout the presentation, you can type your questions into the question box on the control panel. These will be answered at the end of the presentation. Dr. Genet will compile and then pose these questions. At the end of the question and answer period, if there is time, attendees will be able to ask any additional questions. If you have a question, please raise your hand by clicking on the hand icon on the left side of your control panel. You'll be recognized by me, the moderator, and your mic will be unmuted. You will then be able to ask your question live. This webinar is being recorded and archived on the NAVBO website for future use. Dr. Eichmann is an Ensign Professor of the Department of Medicine Cardiology and a Professor of Cellular and Molecular Physiology at the Yale Cardiovascular Research Center. Her laboratory studies vascular and lymphatic development with particular emphasis on mechanisms that direct patterning and guidance. Please welcome Dr. Eichmann.
Okay, so good afternoon, everybody, and thank you very much for joining. We will be talking today about the intestinal villus vasculature, and I will present you some recently published results about lacteal junctions and the control of chylomicron absorption. So to situate you, we will be talking about the small intestine that you can see in this schematic here, and you will see a higher magnification view of the inside of the intestine, the lumen. And so this is composed of thousands of little villi, which are composed by epithelial cells at the border. And here's where nutriment uptake from the gut occurs. Now, within every single one of those villi, we have a centrally located lymphatic terminal vessel called a lacteal. And these are surrounded by arterial and venous blood capillaries, and again surrounded by the intestinal epithelial cells. And so this intestinal vasculature is very important in controlling and distributing the nutriments taken up from the gut. And so small molecules such as sugars and amino acids can be taken up by the blood vessels and will be transported to the liver. But the majority of lipids that are taken up by the epithelial cells here, by the enterocytes, are packaged into large particles called chylomicrons, and these particles are too large to squeeze through the blood vessels and are instead taken up by the lacteals. And this is schematically shown here. A lipid is taken up by enterocytes, packaged into chylomicrons, and these will enter into the lymphatic circulation. Now, once they're inside the lacteals, the chylomicrons will be transported to the mesenteric lymphatic vessels, and from there to the thoracic duct, and then to the subclavian vein, so they will enter the venous circulation close to the heart, and from there will be transported to the liver and actually metabolized. Now, these lacteals were discovered in the 17th century because of their milky white appearance after a meal, and it has so been long recognized that these are the major sites of lipid uptake into the body. But quite surprisingly, the cellular and molecular mechanisms regulating chylomicron entry into the lacteals were actually fairly poorly understood. And it was sort of assumed that these lacteals were passively open and would kind of suck up everything around in a passive manner. And I will show you data today demonstrating that this is not quite true. And the take-home messages from my talk are that there are specialized junctions called button junctions between lacteal endothelial cells that control chylomicron absorption, and that this is by no means passive, but an active mechanism that requires VEGF antagonism throughout life. VEGF, for those of you who do not know, is the major angiogenic growth factor and a multifunctional cytokine actively studied in the angiogenesis community. And I will show you data that demonstrate that if you elevate VEGF levels, you can actually transform these permeable button junctions to impermeable zipper junctions, thereby creating a barrier that inhibits chylomicron uptake. All right, so how did we get into this study? So my laboratory has been investigating for the past 10 years or so a family of guidance molecules shown here. These are called slits, netrins, semaphorins, and ephrins. And these molecules function in guidance of growth cones of developing axons and also in angiogenesis. And they are targets for treatments to promote or inhibit angiogenesis. And among several of these molecules that we have studied, we became particularly interested in one, which is called Nrp1, shown here. This is a powerful regulator of angiogenesis that has also been implicated in metabolism regulation. And so in this paper, published in 2012 by the Ericsson lab, they could show that mice deficient in VEGF-B, when crossed on a diabetic DB-DB background, developed a morbid obesity. So these mice weigh 70 grams, a diabetic mouse weighs 50 grams, and a normal mouse weighs 25 grams. And interestingly, despite their morbid obesity, these VEGF-B mutants remain normal glycemic and failed to develop type 2 diabetes. Now, VEGF-B is one of the ligands for a receptor complex composed of Nrp1 and FLT1 or VEGFR2. And so when this study was published, I wanted to make a receptor mutant for VEGF-B and study such an obese but metabolically happy mouse. And so the model that was proposed in that paper was that VEGF-B binding to this receptor complex would initiate signaling to induce fatty acid import into tissues. An absence of this signaling would lead to lack of fatty acid import and shunting into white adipose tissue, hence explaining the obesity, but enhancing glucose transport into tissues and hence explaining the normal glycemia. Now, the problem with this model is that the FLT1 tyrosine kinase domain does not signal well in most studies, and an alternative model has been proposed in which it was said that FLT1 acts as a decoy receptor. So FLT1 has very high affinity and specific ligands, including VEGF-A, VEGF-B, and PLGF. And so its removal can actually enhance VEGF-A binding and signaling through the main signal transducing receptor, VEGF receptor 2. And this could improve metabolic functions by promoting adipose tissue angiogenesis. And Nrp1 could actually enhance this decoy function. It is expressed at 5-fold higher levels approximately when compared to VEGF receptors in endothelial cells. And the data I will show you today actually support a decoy function for Nrp1 and FLT1 in the intestinal villus vasculature. All right, so we set out to create mice with endothelium-specific and inducible deletions of FLT1 and Nrp1 by taking the flox alleles and crossing them with Ralph Adams' VE-cadherin CRE-ERT2 mouse. And so this allows you to inactivate both receptors in endothelial cells specifically once tamoxifen is administered. And this is how we did the experiment. We let the mice grow up and induced gene deletion between 5 and 6 weeks of age, then waited for 2 weeks, and then put the mice on high-fat diet feeding for a period of 16 weeks. And we get very good gene deletion as shown on this Western blot on mouse lung endothelial cells of Nrp1. This is quantified here. And we also got good deletion of FLT1 as measured by qPCR. And this is the composition of the diet that the mice received. They were on a high-fat diet in which 60% of calories come from fat. Normal diet, only about 10% calories come from fat. And here is the result that we obtained. Rather than getting morbidly obese mice, we actually got thin mice that were resistant to diet-induced obesity. And so this is a new phenotype and the opposite of what was expected from the signaling model. Now here are the weight data. We can see that control litter mates, which are tamoxifen-injected Cre-negative litter mate controls, double their body weight over the 16 weeks of high-fat diet feeding, whereas the Nrp1, FLT1 endothelial mutants gain little weight. If we put these mice on normal chow, we do not see any weight differences between the mutants and the controls. And here are the data from the individual Nrp1 or FLT1 endothelial mutants. So Nrp1 endothelial inducible knockouts gain weight normally on a high-fat diet, whereas FLT1 mutants show a little bit of weight reduction. And this is similar to previously published data from the Alitalo and the Erickson labs, and the cow labs. And so this shows that Nrp1 and FLT1 are unlikely to work in a complex to regulate effects on weight gain, because if they did, you would expect a symmetrical phenotype in the individual mutants. Now, when we open the mice, we can see lots of abdominal fat in the control mice, but little fat in the mutant mice. And when we analyze their body composition, we can see that the double mutants have less body fat, but more lean body mass. And their body composition actually resembles that of control animals on normal chow. Now, the mice do not develop hepatic steatosis, as shown by body piece staining of livers and quantified here. And this even after 13 weeks of high-fat diet feeding. Now, importantly, when we put the mice in metabolic cages and we measure calorie intake or energy expenditure or activity, we can see that the controls and the mutants actually have a similar intake, expenditure, and activity, and their glucose tolerance is improved. And so this tells you that the resistance to diet-induced obesity is independent on changes in any of these parameters. Now, when we look at lipid levels in the blood of the mutants, we can see that plasma triglyceride, cholesterol, and HDL are all decreased. And so overall, that shows you that either the mice have a defect in lipid uptake or they metabolize the lipid much faster. And to test for lipid uptake, we did the following experiment. We took the mice, we fasted them overnight, then gavaged them with olive oil, and took blood at various periods of time and measured plasma triglyceride content. And this is what we got. In control mice, you can see a peak of plasma triglyceride 2 hours after a gavage. And in the mutant mice, you do not see such a peak. If we inhibit plasma lipid reuptake from the plasma by inhibiting lipoprotein lipase, you can see that plasma lipid levels rise much more in the controls, up to 700 milligram per deciliter, but they still fail to rise in the Nrp1, FLT1 mutants. And the same happens when we gavage the mice with radioactive lipid. And so this tells us that the primary defect in these mice is defective lipid uptake. Now, we also looked, at lipid content or calorie content in the feces, and we can see that this was significantly increased in the double mutants, suggesting that the fat is excreted rather than absorbed. However, these measurements show that not all of the absorbed energy is excreted in the feces of the mutants. And indeed, when we do oil red O staining in the distal gut of the controls and of the mutants, we can see significant oil red O accumulation in the distal colon of the mutants. So the lipid is absorbed in the wrong place, and what it does in this situation is currently under investigation. All right, so the data I've showed you so far led us to focus on the site of lipid uptake in the lacteals, of the intestinal vasculature. And so we reasoned if chylomicron uptake into the mesenteric, into the lymphatics was defective, we should be able to see that in postnatal mice. And so these mice drink milk from their mothers, which can be seen in their stomach, and the chylomicron-rich fluid is transported into the mesenteric lymphatics of these mice. And you can see that very easily just by inspecting the baby mice magnification. Now, if we induce neonatal deletion of Nrp1 and Fit1 by giving the mice tamoxifen at 1 to 3 days after birth and looking at a 7-day-old mouse, you can readily see that the content of chyle in the mesenteric lymphatic vessels is much reduced in these mutants. And if we do this with the individual mutants, Nrp1 or Fit1, remember these mice gained weight normally on a high-fat diet. We can see no such defect. And this can be quantified by counting the number of lymphatic vessels filled with chyle, controls, mutants, and the single mutants do not have a defect. And so this is a much faster experiment and was very instrumental in our further research. All right, so we next looked at electron microscopy in the mesenteric villi at the level of the lacteals. And so this is first shown at low magnification here. And so you can see that lacteals are present in both the controls in the mutants, but they appear to be thinner in the mutants. And importantly, there is no content, chylomicron content, that we can detect in the lumen of the mutant lacteals. And here we look at this in higher magnification. You can see lymphatic endothelial cells are present in both the control and the mutants, and they border the lacteals. We can see chylomicrons present on the enterocyte side in both controls and mutants, but the lacteal lumen, which is completely full of chylomicrons in the controls, appears empty in the mutants. And if we look at even higher magnification at the junctions between the lymphatic endothelial cells, we can observe a lot of open junctions in the controls, suggesting chylomicron passage through these junctions, and the closed junctions and the empty lumen in the mutant mice. Now, we also looked at synthesis of key enzymes required for chylomicron assembly in the controls and the mutants and found no difference between them. And so this led us to suppose that the primary defect in these animals could be the failure of the chylomicrons to traverse the lacteal barrier, perhaps via effects on lacteal junctions. And so to look at this in more detail, we did a whole mount immunostaining of lacteals with LiVE1, so this is shown in red here, and the junction marker VE-cadherin, as you can see here. And you will appreciate that in the control animals, VE-cadherin staining of lacteal junctions shows an interrupted pattern of button junctions. I'll show you that in a minute. Whereas the mutants have a lot more straighter junctions, which are called zipper junctions. And quantification by observers blinded to the experimental conditions showed that there were many more such zipper junctions in the lacteals of the mutants. Now, button and zipper junctions were first described by Donald McDonald in a very beautiful paper on Journal of Experimental Medicine in 2010, and they are schematically represented here. So it is known that the initial lymphatics, so the terminal lymphatics in all tissues, have these oak leaf-shaped lymphatic endothelial cells and special junctions in which some areas are VE-cadherin positive, like here, and they will close the junction, but other areas form these flaps, and these will always be open, and they will allow a fluid uptake and also uptake of immune cells. And so these button junctions in the terminals are actually specialized for fluid and immune cell uptake. And in contrast, the collecting lymphatics, which will take the fluid back to the venous circulation, have zipper-like junctions that much resemble the junctions in blood endothelial cells. And so these will prevent the lymph that is taken up from leaking out again. And so this dichotomy between uptake and transport function is really critical for proper lymphatic vessel function. Now, very importantly, the McDonald lab also showed that developing lymphatics prior to birth all have zippers, but not buttons. And so they must acquire these button-like specialization through an as yet poorly understood mechanism. And we reasoned that Nrp1 and Fit1 could participate in this button junction formation. Now, in addition to lymphatic endothelial cells, we also observed a defect in junctions in the blood endothelial cells of the mutants, and this is shown here. So this is an arterial venous capillary loop in a villus, and you can see in the control, VE-cadherin forms these nice straight junctions, whereas in the mutants, the VE-cadherin appears internalized, and so this is a characteristic of vascular leak that is induced by high VEGF signaling. And indeed, if we look for vascular leak by injecting the mice with a mixture of dextran and isolectin before, we can see that there is a lot of leak in the villi of the Nrp1 Fit1 mutants, and this is quantified over here. And so this was suggestive of high VEGF signaling. So we looked for VEGF signaling in the mutant mice by qPCR on intestinal lysates. And you can see here that there is no difference between the expression levels of VEGFs between the controls and the mutants. But you can appreciate that VEGF-A is expressed at highest levels in the intestinal lysates, followed by VEGF-C, which is a lymph angiogenic growth factor, and VEGF-B, the factor that we started out with, is not even expressed in the intestine, ruling out roles for this family member in the phenotype we're discussing today. Now, when we looked at VEGF receptor signaling initiated by VEGF in the controls and the mutants, you can appreciate that activation of intracellular phosphoresidues Y949 and 1173 was much increased in the double mutants compared to the controls, and this is quantified over here. And on this slide, I'm showing you the comparison of the double mutants with the Nrp1 and Fit1 single mutants. And you can appreciate that only the double mutants show this increased VEGFR2 signaling, whereas the individual single mutants do not show that to such an extent. And this suggests that Nrp1 and Fit1 function as a double decoy in the intestine to restrain VEGF signaling through VEGF receptor R2. Now, if increased VEGF signaling were the cause of this phenotype, we should be able to mimic it in a gain of function experiment. And so we tried that by injecting either nothing or VEGF intravenously into mice for 30 minutes. And here we're looking at the blood vessels in the villi, and you can see that VEGF induces junction breakdown. So this was suggested by many previous investigators, very well known that VEGF induces junction opening in blood endothelial cells. What was much more surprising was that if we looked at the same mice at the lacteals in the villi, you can appreciate the button junctions in the uninjected animals, but the VEGF injected animals formed a lot of these straight zipper junctions. So VEGF does the opposite to lymphatic endothelial cell junctions compared to blood endothelial junctions. This is quantified here. Big increase in the zipper junctions by VEGF-A. Now, VEGF-C, which is a ligand for both VEGF receptor 2 and VEGF receptor 3, does this to some extent, but if we inject the mice with VEGF-C 156S, which is a VEGF receptor 3 specific ligand, we got no effect on junction zippering. And so this tells us that VEGF-A signaling through VEGFR2 on lymphatic endothelial cells is likely responsible for this phenotype. Now, if this were the case, we reasoned that we should be able to rescue the chylomicron absorption defects in the Nrp1 Fit1 mutants by blocking VEGFR2 signaling. And we did this following a neonatal gene deletion of Nrp1 and Fit1, and giving these mice a single injection of DC101, which is a VEGFR2 blocking antibody, 4 hours prior to sacrifice. And you can see on this Western blot that this treatment reduces the excessive VEGFR2 signaling seen in the mutants. And very satisfyingly, this treatment rescued the appearance of chyle in the mesenteric lymphatic vessels of the mutants as quantified over here. DC-101 treatment also rescued the zipper junctions in the mutant lacteals, as shown here and quantified over here. More zippers in the mutants, fewer zippers after DC-101 blocking. The DC-101 experiment reduced the increased leak in the mutant animals, as shown here and quantified over here. And if we do this treatment in adult mice, you can see that DC-101 also rescued the appearance of plasma triglyceride in the mutant animals, at least partially. Now, the next important question was, what was the primary site of action of this high VEGF signaling? Was it the blood endothelial cells or was it the lymphatic endothelial cells? And to test that, we used a blocking antibody against VE-cadherin, which is the critical junction modulating enzyme molecule. And so we tested, we interrupted VE-cadherin with a function blocking animal antibody called BV13. And so again, after a neonatal gene deletion of NRP1 and FLT1, we gave a single 4-hour treatment of BV13 followed by analysis of the mice. And as you can appreciate in this panel, if we do this in the NRP1 FLT1 mutant animals, we can rescue the appearance of chyle in the mesenteric lymphatic vessels, as quantified here and here, without BV13, with BV13. But if we do this treatment in control animals, there's absolutely no effect on chyle uptake. So these two graphs here. Now, if we look at the junctions in the blood vessels of these animals, you can see that the BV13 treatment disrupts junctions in the control animals. See, compare here to here. Much like we see disrupted junctions in these mutants. But remember, BV13 had absolutely no effect on chyle uptake in the control mice. In contrast, if we look at the lymphatic junctions, we can see the zipper junctions in the lacteals of the mutants without treatment. But if we treat with BV13, we can disrupt these junctions. And so this suggests to us that the primary effect of this treatment and of the rescue is by disrupting these lacteal zipper-like junctions. Okay, now there is very little known about modulation of lymphatic endothelial cell-cell junctions, but this process has been very intensely studied in blood endothelial cells. And so it is known that junction opening in blood endothelial cells involves modulation of the VE-cadherin cytoplasmic tail, and unfolding of this tail and coupling to cytoplasmic F-actin, and this creates a pulling force that helps the junctions to open. And this force is dependent on active Rho and Rho kinase, and so we wondered if a similar tension helped these button junctions to remain open, and if so, could we block this process using a Rho kinase inhibitor, ROCK inhibitor. And so to test that, we used neonatal wild-type mice, and again, we gave them a 4-hour treatment with the Rho kinase inhibitor Y27632. And you can appreciate that if you do this, we can actually prevent chyle uptake into the mesenteric lymphatic vessels of wild-type mice. We can change the morphology of the lacteal junctions from more interrupted to more zipper-like, as quantified over here. And if we do electron microscopy studies of such treated mice, you can see that the appearance of chylomicrons in the lacteal lumen is inhibited, and many of these junctions are now closed instead of being open in the controls. And so overall, this brings us to our model. We believe that in the intact villus, blood vessel junctions are closed and lacteal junctions are open, thereby allowing chylomicron uptake and fat absorption. And in the NRP1 FLT1 mutant, this situation is reversed. Blood endothelial junctions are open, lymphatic endothelial junctions are closed, thereby preventing chylomicron absorption and conferring resistance to diet-induced obesity. And so what we think is happening at the molecular level is that NRP1 and FLT1 cooperate in the villi to function as tonic VEGF decoys, thereby decreasing back permeability and allowing leg button junction formation. And how is this process regulated developmentally? We believe that the process is actually initiated by the chylomicrons themselves. So Karina Yaniv several years ago demonstrated that ApoB can directly bind to endothelial cells and increase the expression of FLT1. And we have confirmed these results in the intestine as well. And so this tells us that as soon as chylomicrons are produced, they initiate expression of this decoy receptor, thereby allowing leg button junction formation, chylomicron uptake, and guaranteeing a survival of the mouse. And so I've shown you that blocking of Nrp1 or FLT1, increasing VEGF levels, or ROCK inhibition all lead to formation of leg zipper-like junctions, failure of chylomicron uptake, and resistance to diet-induced obesity. And better understanding of the modulation of these lymphatic endothelial cell junctions might lead to novel approaches to combat diet-induced obesity and lead to plasma lipid lowering. And so I'm at the end, and the most important slide is to thank all the people who did the work. In particular, the lion's share of the work was done by Feng Zhang, a very talented postdoc in the lab, with help from many other people, including Gael, who will take your questions today. I would also like to thank a lot of people who provided us with mice and help from metabolic collaborators, Bill and Carlos at Yale, and the Yale Mouse Metabolic Phenotyping Center and Jerry Schulman for their help with the data analysis and interpretation. Thank you very much for your attention. I'll be happy to take questions.
Thank you, Dr. Eichmann. I see we have several questions from the audience. I'm going to turn this over to Dr. Genet so that he can direct these questions to Dr. Eichmann.
Okay, thank you very much. So we have a question about VEGFR2 expression in the lacteal. So do you know if VEGFR2 is expressed in the lacteal?
Yeah, so we've done immunostaining of VEGFR2, and we can detect a clear immunosignal on the lacteal endothelial cells, and this is shown in one of the supplementary figures in the paper.
And do you know if other growth factors can change the junction in the lacteal?
Yeah, that's a great question. We don't know yet, but we're testing this currently. So I showed you that VEGF-C can induce the closing of zippering of these junctions to some extent, but less well than VEGF. In terms of other growth factors, angiopoietin would be another candidate to modulate these junctions, but we haven't investigated that yet. And we haven't seen any changes in the angiopoietin 2 levels in our mutant animals.
So we have another question about the cell survival. As VEGF-A regulates cell survival, do you see any change in your mutant mice about the survival of endothelial cell in the lacteals?
Yeah, that's another great question. So we've looked at these, so many of the acute experiments I've shown you were done with a very short-term treatment for VEGF. But we've also looked at the mutant mice, the Nrp1, FLT1 mutant mice, at many ages postnatally and also in the adult, and after prolonged high-fat diet feeding up to 8 months, I believe. And we have not observed any changes in the length of the lacteals, or even in the health of the intestinal epithelium as a whole. And this is a little bit surprising, really, when you think about the situation I've presented. There should be very significant edema, there might be inflammation, and despite all this, what we've seen is that the mice appear to tolerate this Nrp1 FLT1 deletion quite well. And so this tells us that perhaps these zipper junctions are actually quite capable of absorbing fluid and maybe also immune cells, although we don't know about the inflammatory situation that much so far, except morphologically nothing seems to be very much amiss in the mutant mice. I hope that answers.
Okay, so we have another question about the specificity of this phenotype in the guts. So do you see other lymphatic change in other capillary beds other than the guts?
So that's another very good question. And we were asked to do these experiments. And so we looked in the Nrp1 and other lymphatic beds, in particular in the dermal lymphatics. That's where we've done the most extensive analysis. And we have not seen any defect in the dermal lymphatics of these mice. We have sort of less complete data in the trachea and the diaphragm and have not seen any problems with the lymphatic junctions there. And in contrast to that, we have also done the VEGF overexpression experiments and looked in the dermal, tracheal, and diaphragm lymphatic junctions. And there we have seen that VEGF is able to induce junction zippering. So the VEGF effect appears to hold throughout the animal, whereas the VEGF antagonism through Nrp1 and Fit1 appears to be intestinal specific. The reason for that might have to do with Fit1 levels and the constant feedback provided by the chylomicrons, which increase its expression, or it could have to do with the other mechanism and other cell types that produce these receptors, such as macrophages.
Okay. Do you know if obesity can change this transition between button and zipper, the obesity itself?
I do not, and I think that's a very important question. I do not. I do not know.
Okay. Okay, so maybe a last question about what is the source of the VEGF?
All right, so VEGF expression in the intestine was examined by various investigators, and it appears to be coming from different sources. So these include, I believe, the smooth muscle cells, the macrophages, and there is also VEGF coming from the mother's milk. So there are various sources that are a candidate for VEGF production, and which one is the relevant one or the biologically relevant one, we don't know yet. It's also kind of complicated experiment to address.
Okay. So we don't have any more questions about from the attendees. So thank you very much.
Okay, thank you both. If there are any other questions, please raise your hand. Once I recognize you, please state your question. Okay, I don't see anyone raising their hand, so thank you both. And that was a wonderful presentation. I hope you found this information beneficial. These webinars are brought to you by the NAVBO Education Committee. Our next two webinars will also be from speakers at Vascular Biology 2018, held recently in Newport. Please join us in December and January for talks by Tim Law from Boston Children's and Josh Hutchison from Florida International University. Dates will be announced in an upcoming newsletter. Please fill out the evaluation form at the end of the webinar or when you receive it in a subsequent email, and let us know what you thought of the webinar and any topics you might want to see in a NAVBO webinar presentation. Thank you.
Thank you.